The following audio recording has been designed for learning purposes only. Do not share, distribute, or provide any type of copies, or this audio lesson will not be made available to you. Good luck with today's lesson.
All right, guys, we are going to go ahead and get started. During lecture today, we're going to finish chapter 9, tackle a teeny tiny part only of chapter 31. We're going to skip most of it. And then we're also going to get into chapter 34, get however far we get with that chapter. Of course, the big thing happening today is the lab practical. So the first thing I want to ask you guys is if you have any lab questions that you want to go over. I know that's probably like in the forefront of your mind, so any lab stuff you want to ask about? No. Okay. If a question pops into your head about lab at any point, I don't care what I'm talking about, just interrupt me, say lab question, and I'll come right to you. I want to make sure you guys feel as good as possible before we head over to lab. In the meantime, if not, we'll go ahead and finish off chapter 9. I cannot remember exactly where we left off. Did we go through any of the steps of mitosis?
Yeah, we left off at cytokinesis.
Oh, perfect. So we went through all the steps of mitosis.
I believe.
Does that sound familiar to everybody else? Okay, cool. Thank you guys so much. What I want to start with then, because I don't think we did yet, I want to watch video to hopefully pull it all together for us. And it'll be a really nice way, I hope, of reviewing all the steps that we went over last time. So before I show the video, maybe let's just get ourselves back into the fold here. Remember, what chapter 9 is focused on is the process of the cell cycle. That's the process that happens in our cells when we are trying to make a new cell. We'll refocus in just a couple of minutes about why we would want to be doing this process, like when and why and where do we need to make new cells in our body. But all new cells, except for sperm cells and egg cells, all new cells are made by this process. So our kind of big picture that we focused on right away for chapter 9 is our overall idea of what's happening. We take one cell, whatever kind of cell we're starting with, we're going to watch it during interphase get bigger, make more of everything inside of it, and then what we tracked with mitosis and cytokinesis is how we then split that one parent cell into two identical daughter cells. Some key points I tried to get across on Monday are to recognize that you are making identical daughter cells. They're like carbon copies of each other. They're both exactly the same as the parent cell. And that's why I always try to say, like, hey, whatever cell you start with, good, bad, or ugly, whatever you start with, that is exactly what you are making. That's where we talked about the importance of those checkpoints. Remember those little 3 times around the cell cycle where the cell kind of pumps the brakes for a second, just puts a pause on everything, and just takes stock of the situation. Is everything going okay? Is the cell damaged? Are we able to proceed? You hope everything looks great and you can continue through the process. Or you hope if there is a problem, you recognize that there's a problem and try to fix it. If you can fix it, great, go ahead and fix it and move on. But if there's a major problem that cannot be fixed, you do not want to keep going because you're essentially then going to make a cell that's screwed up that could lead to more problems eventually later on. And so if that's the case, if there's a big problem that can't be fixed, that's when we would hope that apoptosis would be triggered. Remember, apoptosis is that programmed cell death where a cell is kind of just hitting a little self-destruct button, taking itself out of the equation because it has recognized, like, hey, something's wrong with me, I'm a pretty messed up cell, and I shouldn't be here causing additional problems. So that cell would commit to undergoing apoptosis, and we wouldn't worry about it anymore. Remember, apoptosis might sound bad. Our cell is killing itself. That's a good, healthy process that we want to be able to do. And in fact, if you can't do it when you should be able to, that's when some problems can arise. I'm going to close that door so we're not hearing all that noise from the hallway. Once we established, how important it is that this process goes properly, and the fact that we had to do some prep work to get ready to actually go through the division process, after we got a whole bunch of vocab, remember what I said about the vocab? You got to get it down. Chromatin, chromosome, diploid, haploid, chromatid, centromere, kinetochore, centrosome, centriole, all those words, we learned them on Monday. we saw how they're being used on Monday to describe the process and the structures that we saw throughout mitosis. So just remember to really key into those vocab words. That's not all you're going to have to know, but without understanding the vocab, you're going to have a hard time tracking the process. So what I want to do is have us sort of officially start, I guess, after that quick rehash, by watching the video that I had mentioned, just to give us an overview of the steps that we saw on Monday. Before I start the video, do you guys have any questions about the steps that we saw Monday or the overall kind of point of mitosis? Okay, I'm going to switch this over to the desktop then, and we'll watch this video. I like this one. Even though it's like the older graphics and the voice is sort of robot-y, I think it does a good job of showing a lot of the detail that we went over. All right.
During interphase of the cell cycle, the genetic material of the cell is found in the form of chromatin.
I'm going to pause it real quick. You guys see the problem there?
Yeah,
It's labeled chromosome. Like, is that a chromosome yet? No. They correctly said chromatin, which is great, and then they put the wrong label. That like drives me crazy, clearly. So I just wanted to pause it and make sure you guys recognize that that's not yet correct. It's not chromosomes until we started mitosis.
And located within the nucleus of the cell, which is surrounded by the nuclear envelope. During the S stage of interphase, DNA replication takes place, and thus the cell leaving interphase and entering mitosis already has replicated its genetic information. In addition to the replicated genetic information, interphase cells that are about to divide also replicate their centrosomes. In animal cells, these centrosomes are formed from centriole pairs. As cells leave interphase and begin prophase, the first stage of mitosis, the chromosomes begin to condense. The nuclear envelope begins to vesiculate,
Or break
and
down.
the centrosomes migrate to opposite ends of the cells to define the two poles. During prophase, the mitotic spindle apparatus begins to form by the polymerization of tubulin proteins. This creates microtubules that emanate from each centrosome. Chromosomes completely condense such that sister chromatids formed from the replicated genetic information and joined at their centromeres are clearly visible. During prometaphase, the nuclear envelope completely breaks down and the mitotic spindle is formed. Polar microtubules extend from both poles and overlap with each other.
I'm going to pause it just for a second because I think in some of the figures from the book and in this video, they are referencing two different kinds of spindle fibers or microtubules. We just called it all spindle. So I just want to kind of make the connection about why they're doing that. We're going to see in just a second that spindle fibers can be doing one of two things. Sometimes they're going to be grabbing onto the chromosome. That's sort of how we focused on them on Monday. The fact that in just a second we're going to see the spindle fiber grab onto that kinetochore protein. The parts of the spindle that actually grab the kinetochore protein on the chromosome, those are sometimes called kinetochore spindle fibers to kind of remind you what they're doing. Other spindle fibers, and I don't know which one it's going to be, so I'll just point to this one for right now. If you watch what they're doing, they actually are not touching a chromosome at all. Instead, those two spindle fibers are going to interact with each other, and when we start to push the cell apart, they push off of each other to help shove the cell further apart. Since they're not touching a chromosome, we're not going to call them kinetochore spindle fibers. So to differentiate them, that's where this term that they just mentioned, polar microtubules or polar spindle fibers, it just means they're coming from the poles to meet up with each other in the middle. No chromosome touching involved. I usually just call it all spindle because it is, but I wanted to make sure to explain the difference in those terms in case you see it in the book or if you're watching the video back so that it wouldn't be more confusing.
The sister chromatids become attached to the mitotic spindle via kinetochore microtubules that attach to the kinetochores, which are bound to the centromeres. As prometaphase is nearing its end, the sister chromatids are observed to jerk back and forth between the two poles. During metaphase, the sister chromatids become localized in a region called the metaphase plate. Once the chromosomes have aligned at the metaphase plate, metaphase is complete. When metaphase is completed, anaphase begins. The sister chromatids separate, and each chromatid is now linked to only one pole via a kinetochore microtubule. The kinetochore microtubules shorten, and the individual chromatids, now called chromosomes, are drawn to the poles. Toward the end of anaphase, the polar microtubules push against each other and cause the two poles to be moved farther away from each other. Each daughter cell will receive the same complement of chromosomes as was originally found in the mother cell, thus assuring that each daughter cell gets an identical copy of genetic information. During telophase, the chromosomes decondense and most of the microtubules depolymerize. Portions of nuclear membranes surround each individual chromosome, creating vesicles. The vesicles fuse with each other to create a nucleus containing all of the chromosomes at one pole of the cell. In animal cells, cytokinesis, or cell division, involves the formation of a cleavage furrow.
I'm going to pause it again just to make a point. In the video, they just kind of matched what the figure in the book did, the very early figure where they explained all of mitosis, and now they're going to add in cytokinesis. Remember what I stressed on Monday? Cytokinesis actually starts during anaphase. They didn't show you the little pinch in, the cleavage furrow starting in anaphase in this video, but again, that's what we are counting it as. Anaphase is when cytokinesis begins.
The plasma membrane is observed to constrict and eventually separates the cell into two daughter cells. The daughter cells then enter interphase, where growth and enlargement occurs, leading to either preparation for another mitosis or differentiation and specialization occurs.
All right, so sorry for the interruptions along the way, but I wanted to point out some important stuff to you guys. Do you have questions about the video?
The microtubule organizing center, is that the centriole or the central zone?
Both, right? So remember, centriole, the two of them together is the centrosome. So technically speaking, the centrosome is the MTOC, but it's made of centrioles. So that's one where I'm okay with a little bit of flip-flopping because either one is technically correct. Yeah. Other questions? Okay, I wanted to draw your attention to the last thing they said in this video. These daughter cells that we've just made, they're kind of pointing out what's going to happen with them. Okay, so they're immediately, like as soon as they let go of each other, they're immediately considered to be in interphase in the next round of the cell cycle. Remember we talked about that little blip off the main figure, G0? If the cells are not going to actively divide, they're still technically in interphase, but we'd flag them as G0, like, hey, they're not gearing up, they're not getting bigger, they're just doing their thing. That's this whole idea of differentiation and specialization. That just means the cell is going to be whatever kind of cell we made it to be. So if we just made a new liver cell, it's going to kick it into high liver cell gear and just start participating in whatever the liver cell is supposed to be doing. If it's a kidney cell, it starts behaving like the kidney cell we made it to be. In other cases, in other circumstances, we might need more of those cells right now. So if we're trying to make a whole bunch of new cells, as soon as we make this and it's considered to be an interphase, it would swing right into G1, SG2, and go through another round of mitosis. Again, this comes back to the whole point that cells are supposed to be picking up on signals around them to tell them what to do, and that's how they would come to this decision, based on what they're being told. Any questions about that? Yeah.
Would you just explain to her? I'm sorry.
Oh, so she asked about the difference between centrosome and centrioles. Okay. So she asked about the MTOC. That stands for Microtubule Organizing Center. And that, remember, is like the boss of all the microtubules that are making up the spindle. And those two terms, centrosome and centriole, there's a little overlap there because technically the centrosome, it's made up of those two things. The centrosome is the MTOC, but each one of those lines are centrioles.
Oh, okay.
So you could say that the MTOC is made of centrioles, or you could just refer to it as the centrosome.
Okay.
Either one is okay.
All right.
Yeah. Yet another fun example of where there are multiple words for the same thing.
Yeah.
So it's, I know it's a little confusing there. Go ahead, Maya.
Lab question.
Yes, go for it.
Should we know the formulas for converting between Celsius and
Hell no, you don't need to know temperature conversions. No temperature conversions. You will be moving some decimals in the swooping kind of deal. I think I put a couple of those on there, but I will not make you ever do temperature conversions, especially when you don't get a calculator.
Thanks.
Yes, good, yep. Any other lab questions while we're on it? Okay, so I'm gonna flip it back to the laptop, and I wanna talk just a little bit more about cytokinesis, just to hammer home a couple of points. You already know what this is, right? Remember, mitosis is specifically looking at how the nucleus and its contents, all those chromosomes, are being divvied up between the daughters. But you guys know the nucleus is not the only part in a cell. We also have to divide the cytoplasm, the mitochondria, the ER, all that other stuff. And that's really what we're doing during cytokinesis. This, as I've stressed several times, wanted to make sure I put it in here again officially, this process of the cytoplasm starting to be divided, that starts during anaphase. So it is ongoing, it is overlapping with the end of mitosis. Starts in anaphase, continues throughout telophase, and the process of cytokinesis really isn't done until the instant when those two daughters officially let go of each other. That signals the end of cytokinesis, it signals the end of that round of the cell cycle. Now, we've really only been looking at animal cells. That's what the diagrams that we worked our way through at the end of class on Monday really were highlighting. The video we just watched was about an animal cell as well. So I wanted to just make sure we came back to a point I snuck in on Monday, that due to the structural differences that exist between animal cells and plant cells, there have to be slight differences in how cytokinesis plays out, right? And I think we can probably get here without too much of a stretch. We've seen an animal cell like just pinching in and essentially peeling apart. Remember, animal cells do not have a cell wall, so we're more pliable essentially. Plant cells have that rigid cell wall. So do you think a plant cell can just squeeze in and pull apart? They can't. It would be harder for them to do that because the cell wall is so tough, you can't just squeeze it in and pull it. So plant cells have to tackle this process in a different way. We're going to get a closer view of animal cells first. The book has kind of more detail here than what I want you to worry about. Really why I'm showing you this slide is to remind you what the cleavage furrow is. So keep that. Another reminder that this process starts in anaphase. We're not going to worry about this, okay? So if you have slides printed, scribble off that last bullet point. I just really like to show this slide because of the picture. I think it's a cool picture to help us visualize what's going on. So we've got some diagrams here, along with what I think are some really cool microscope images of an animal cell actually going through this process. So what happens with that cleavage furrow is it just gets deeper and deeper, and the cells are continuing to like push off of each other until they eventually kind of snap apart. So right now, this would still count as technically one cell, but we're almost there. As soon as that little bridge is broken, then we've got our two daughter cells, which are both then considered to be in interphase for that next round of the cell cycle, whenever it might take place. The book points out that actin is involved in the process. Again, we're not going to worry about all of that. Any questions on the fact that animal cells form a cleavage furrow as cytokinesis occurs? Okay, so let's look at what plant cells do instead. You already know that they cannot form a cleavage furrow because you can't just pinch a plant cell in. Again, they're a rigid cell wall that helps them maintain the proper shape. It doesn't allow for that process to occur. So we have to deal with this in a different way. Mitosis, the act of dividing the chromosomes, all that kind of plays out the same way. It's just when we get to the final separation of those two daughter cells, we have to do things a bit differently. Instead of pinching and pulling the cells apart, what we're going to do is build a new cell wall from scratch right in the middle to ultimately separate our two daughters. Okay, so we are going to take advantage of our knowledge of a few components that we've learned about previously, right? Number one, cellulose. That's way back in chapter 3, but hopefully you guys remember cellulose is the structural polysaccharide that cell walls are made of, right? It's a big sugar molecule that helps the cell wall be so strong. So if the plant cell is tasked with building a new cell wall, it's going to be making a bunch of cellulose. And then the next job is that we have to put all the cellulose that's made in the right place. Essentially, we have to, here's nucleus of daughter cell 1, nucleus of daughter cell 2. We got to drop it right in between those two nuclei that are forming during telophase. How do we do that? We load the cellulose up into vesicles, our old friend the vesicle, just a whole bunch of little bubbles full of these big sugar molecules, and we send them all right to the middle of the cell. This is the same place where that metaphase plate would have been, like where the chromosomes would have been lined up. They clearly have already been split, but right in that same middle of the cell, all these vesicles start showing up with cellulose. And you guys know what happens whenever we have two little things surrounded by membrane. They're like bubbles, right? They get close together and they merge. So we bring two together, we add another one, and pretty soon we've got this whole big strip of cell wall. And what was the vesicle lipids, they're going to be now on either side, and that makes the new cell membrane to formally separate those two cells and that one cell into two. I got there. Any questions about what a cell plate is, or the fact that this is the structure that plants use in order to complete cytokinesis? Okay, there's a quick little recap video for this one too that I've dropped onto Canvas, and I will post the links later because I know I mentioned this on Monday. I know not everyone can see these videos in their current form, so I'll drop those links today or tomorrow.
In animal cells, cytokinesis completes
Sorry,
cell
guys.
division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells.
Any questions on the video? Okay. So that's the cell cycle. We've covered every part of it now. We know what should be happening, right? Recap. We start with any kind of cell in the body that we need to make more of, except a sperm cell or egg cell. Any kind of cell. Skin cell, brain cell, kidney cell, liver cell, you get the idea, any kind of cell. When we need to make more of it, certain messages are sent to that cell to tell it, like, hey, we need more of you. Start this process of getting ready to divide. That's what would trigger a cell to go from G0, where it's sitting there doing its thing, into entering G1, S, and G2. Remember, the whole idea of interphase is the cell is gearing up and getting ready to divide. How and what is it doing to get ready? The cell, the parent cell, has to get bigger, and it's going to make more of every single thing it has inside of it. You see more cytoplasm, which helps get bigger. You see more membrane being made to allow the cell to get bigger, more mitochondria, more ribosomes, another centrosome. All the parts are being doubled, and importantly, during the S stage, all of the DNA is also being duplicated. Remember why all of this is so important? When we ultimately split into two daughter cells, we want to make sure we're giving both of those daughter cells everything they need to be a successful, healthy cell. We got to give them all the parts they need, and we got to give them the instructions on how to use those parts. So we had to copy everything during interphase. And once we're bigger and bulkier and have more of everything, then the cell is ready to transition into mitosis and cytokinesis. Remember, this part of the cell cycle is the actual division process. So with mitosis, we tracked how the chromosomes are very carefully and meticulously lined up right down the middle and ripped in half so we can guarantee that each daughter cell gets one copy of every chromosome. That's important because those are the instructions for how to do everything. As that process is kind of continuing and wrapping up, we then start dividing all the other assets in the cell too. And by the end of it, we will hopefully have successfully split from one parent cell into two identical daughter cells. Now that we've been through all the steps that are involved, I think it's really important to kind of circle back to why are we doing this? Why do we and other organisms like us, meaning eukaryotic and multicellular, why do organisms like us do this process? Why would we need to make more cells? For us and other organisms like us, it's really for one of two reasons. Either to get bigger, right? And to me, I always think about this in the extreme. All of us started off as a single cell that resulted from fertilization, right? The sperm cell and the egg cell, that one first little cell was us. And I always say, like, you know, babies are pretty small when they're born, but they're already made of millions of cells. So how do you go from being made of one cell to being made of millions of cells? You do a whole bunch of the cell cycle. You do a whole bunch of mitosis. First, we split to 2 cells, then we split to 4. Ramp it up till you've got millions of cells. We don't stop growing when we're born, obviously, but embryonic growth, and then when you're a baby, toddler, whatever, that's our most rapid period of growth. We still have to continue doing mitosis or the cell cycle as we're, mature stature though, because we have to be able to fix issues as they pop up. We have to make new cells to replace old ones that aren't working very well. Or if some of your tissues or cells get damaged due to illness or injury, you gotta be able to make replacements to take over the job not being done by those cells that died. So again, for us, this act of cell division is all about these two things. Either to get bigger by making more cells, or to fix a problem that arose due to cell death or injury, whatever. Any questions about why we do mitosis? All right, now why I keep stressing we and us and organisms like us? You guys know very well that not all organisms on the planet are like us. There are some organisms that are unicellular. There are some organisms that are prokaryotic. And all organisms are made of cells, absolutely, but being made of different kinds of cells or different numbers of cells, that has implications or consequences in terms of why you would go through this process and what it would look like for a cell like you, okay, or not like you. So I think the first thing I want to say is, remember, mitosis is defined as division of the nucleus, right? So technically speaking, mitosis can only happen in eukaryotic organisms. Are bacteria eukaryotic? No, bacteria don't have a nucleus. So do bacteria do mitosis? No, they don't have a nucleus. They can't possibly be doing this technical definition, mitosis, yeah.
Mitochondria can split in two, what you're saying? Or?
Nope, mitochondria do not split in two. At least not that we've looked at. We're talking the whole entire cell has to split in two, yeah. Yeah, mitochondria do have to be, we have to be able to make more mitochondria, and we have not, nor will we ever look at how that process happens. Yeah, so I don't even want to go there. But I'm just trying to raise the point that all cells make more of themselves, but they might do it in different ways. Here comes a big chunk of the chapter that we're going to skip, which I'm kind of sad about, honestly. I really like doing this part of the chapter on cancer because it's very relevant and applicable, unfortunately, sometimes. But I like talking about it because we just spent, quite a bit of time talking about what our cells should be doing. And looking at cancer basically says you're looking at, well, what happens when that process goes wrong? We're going to skip this whole section. So if you have slides printed, multiple slides are about to get crossed off, okay? Anything that says cancer on it, we're getting rid of all of this, okay? So cell cycle gone wrong, cross off, not coming back, not on the test. Not going to go over how cancer cells are different from healthy cells. So that's 1, 2, 3 different slides, crossed off, gone, not coming back, not on the test. Cross off the summary table, not coming back, not on the test. Oh, I'm so sad. Cross off the figure, not coming back, not on the test. Cross off what kinds of issues could lead us down that road, not coming back to it, not on the test. Cross off the crazy figure, not coming back to it, not on the test. And then we're picking it back up here at prokaryotic cell division. I've never skipped that part of this chapter before. That's why I'm so kind of sad about it, but we are on a limited time frame, and that is information that we can let go of and still focus on central aspects of cell biology, looking at how the division process should occur. If you are interested in talking more about the information we skipped just for personal interest reasons, you can come talk to me, I'll walk you through it, whatever, but it's not anything you have to know for exam 3. Okay. Any questions about that stuff that we're skipping, which lightens your load essentially for exam 3. Okay, so what we've got left to do here at the end of chapter 9 is come back to that point I was making before all that cross off, is that not everybody is like us, right? And we're going to talk first about bacteria and other prokaryotic organisms. You know that bacteria are not the only prokaryotes, but they're the only ones we think about in this class. So I'm just going to start saying bacteria. Bacteria, as you guys know, are also made of cells, and their cells also go through a process of cell division. But their process looks different and is used for a different purpose. They don't use cell division to get bigger or to, make one new cell to fix an issue. When they make one new cell, because they're unicellular, right? When they make one new cell, they haven't just made one new cell, they've made a whole new member of their species. So things that are unicellular, like bacteria, when they make a new cell, they're reproducing. Obviously, there's no sex involved in just splitting in half. So it's a form of asexual reproduction. This is the first time in this class that we've really talked about the fact that there, at least in a mention kind of a way, that there are different types of reproduction, right? Obviously, we reproduce sexually. Things like bacteria reproduce asexually. And I'm pointing this out now because it's going to become something that we focus on even more in the next couple of chapters and also in lab. After we do our rat dissection, we'll do an entire lab looking at various means of asexual reproduction, and then another lab looking at various means of sexual reproduction. So this is going to become an idea that we're focused on for the next, couple of weeks in the class. Those babies, those little bacteria babies, if you will, that are being made as a result of asexual reproduction, they're exactly the same as the parents. So even though this process called binary fission that we're going to track bacteria using to make more cells, even though the process looks a little bit different because the cells look a little bit different, the gist of it is basically the same. Whatever cell you start with just gets bigger and then makes more of everything inside of it and then splits in half. Those offspring are exactly the same as the parent was. And that's an idea we're going to come back to as we get deeper into the whole idea of asexual versus sexual reproduction. There's no one perfect strategy. There are different pros and cons associated with each. And again, we'll talk more about that in later chapters. So we're going to track how bacteria go through cell division using this process called binary fission. It's going to look a little different than mitosis. We're not going into nearly as much detail. I think the big point I want to raise is that even though it's not mitosis because there's no nucleus, the gist of the process is the same. We're going to see these bacteria, which typically, there are some exceptions, but typically they only have one chromosome. They're going to do the same thing with it that we did with all 46 of ours. They have to make a copy of it because just like for us, for them, that chromosome contains all the information they need to survive. So they have to make a copy of it to give one to each daughter cell, just like we did with all of ours during mitosis. So we're going to look at a quick figure on the next slide. And again, you can read the descriptions off to the side. I want you to just focus on the picture with you guys because I think it gives us everything we need to know. In that bacterial cell we're starting with, in the parent cell, way up top, you can see that one red squiggle. That's the single chromosome in that parent cell. The first thing that happens is, it's kind of hard to see because now we've got purple on purple. That drives me nuts. But you can see we've made another chromosome. We copied it, just like we did with our chromosomes during interphase. Once we've got 2 chromosomes, you can already tell the cell is getting a little bigger. You can see it's going to pinch in the middle, and we've purposefully separated the chromosomes into the 2 different daughter cells. Even though the structures look different, we're not seeing as much detail here. Really what I connect with on this slide is it's the same basic process as what we saw with mitosis. Cell makes more chromosome, gets bigger, splits in half. Done. Any questions about this process of binary fission that things like bacteria use to make new cells? Okay. So I want to connect back with something I've already said, thinking about why they do this process. Bacteria are, of course, only one example of a unicellular organism. That's a vocab term I know you guys know. It's from chapter 1. It's also been in your lab book, so you hopefully have seen it while you're studying. That applies to bacteria, but it also applies to some things with a nucleus that you've seen in lab, like amoeba, paramecium, euglena, whatever. Any kind of single-celled organism, when they make a new cell, they're not just making a new cell, they're making a new member of their species, right? So what we're looking at here is the purpose behind cell division. Remember, for us, and we'll get a refresher in just a second, for us, it's all about growth and repair, right? When I make one new cell, I don't make a whole new me. I made one new kidney cell or whatever. But when anything that's a single-celled organism goes through cell division, now we've got a new member of the species. This is a form of reproduction. Now, I like that they're showing us this figure. We just saw bacteria on the last slide. What's that right there? A nucleus, right? So it's something like an amoeba. They're driving home the point that I made up here, that not all single-celled organisms are bacteria. If something with a nucleus goes through cell division, then you can call it mitosis, right? So this would be mitosis because this organism has a nucleus, but because it's single-celled, it will be serving the purpose of reproduction, right? So there are kind of two questions you have to ask yourself. Is the organism unicellular or multicellular? That gets at the purpose for the division. And then you ask, is there a nucleus or not? And that gets at what kind of process it would be, mitosis versus not. We're going to see the counterpoint now on the next slide to remind ourselves what we do, which I know we've already discussed. Anything that's like us, meaning multicellular and eukaryotic, the form of cell division we're going to do is mitosis because we have a nucleus. And why are we doing this? Again, growth and repair. I like this figure as a contrast to the previous slide because, right, if I go from 1 cell to 2 or from 3 to 6 or whatever, if I make one new kidney cell, do I make a whole new me? No, it's clearly not a form of reproduction. Again, for us, growth and repair. I really like the table on the next slide because I think it's a really good kind of pointed summary at the issues that I've raised over the last couple of slides, that there are two factors that you have to consider when you're thinking about cell division. What do you call it and what purpose does it serve, right? So look at what we've got over here. Prokaryotes that are single-celled, and then eukaryotes that have a nucleus but could either be single-celled or multicellular, right? So the distinction about what you call the process comes down to, is there a nucleus or not? If there's no nucleus, the version of this process that we've seen was binary fission that bacteria do. If there is a nucleus, regardless of who has it, if there's a nucleus, you call division mitosis. What we've also focused on is, like, why is the cell dividing? What does that accomplish? This is where it doesn't matter really if the cell has a nucleus or not. It matters that it is unicellular. Again, anything made of a single cell, when it makes a new cell, it makes a whole new organism. Those kinds of organisms are doing cell division as a form of reproduction. Versus, again, with us, growth and repair. It's the big two things we're going for. I like this table a lot, again, because it gets right to the heart of the matter. The two questions you have to ask: is there a nucleus? Is the organism unicellular or multicellular? Any questions on this distinction? Okay, so that is it for chapter 9 then. We skipped this whole chunk on cancer, which might not have seemed like that much. It was, probably 6 or 7 slides, I'm guessing. I didn't count. But that usually takes me close to an hour to get through, so we just saved a significant amount of time, which I'm good with. That's the whole point. We're trying to squeeze some stuff into a shorter time period. Before I open up chapter 31, does anybody have questions about the cell cycle that our cells do for growth and repair, or the fact that other kinds of organisms might do a different form of cell division for a different reason? Okay. Chapter 31, we are also going to be skipping significant portions of, okay? Significant, like almost all of it, we're going to skip. Chapter 31, I'm okay doing that because this chapter actually isn't on the official list of chapters we have to do. I always like to include it because it helps us transition. We've spent so much time thinking about cells and what they do, And it helps us kind of follow this pathway of how our cells are organized in our bodies. So I usually do like an abbreviated version of the chapter anyhow, but we're gonna skip pretty much all of this. And I can't show you, I should have pulled it up, maybe I will pull it up on the desktop and show you real quick. Just so that you can see a view of all the slides we're skipping to make this easier for yourself later, hopefully. If I open up the slides here, because if I just jump to the one that we're starting with, I know people who have slides printed might find that a little confusing. We're not going to start until it talks about scaling up to the organ level. So, sorry for the delay. All right, everything from, well, I guess the outline you can leave if you want to, but we're skipping everything from types of tissue to following neuroglia. So that's the first, what, like 17, 18 slides just gone. Okay, so if you don't have slides printed, you can make a note of it if you want. Start chapter 31 at the slide from tissues to organs. All the rest of that goes away. Not coming back to it, not on the test, don't have to worry about it, okay? What that part of the chapter would have represented, quick overview, except it doesn't actually go quickly when I'm talking about it, it takes a while. It would have been a tip of the iceberg kind of view of the different kinds of tissue we have in our bodies and what those kinds of tissues are doing. Instead, what we're gonna do, I'll do it on my laptop, I suppose, we're gonna start by just acknowledging maybe what a tissue is, which you guys already know. Maybe jot it down, I guess, as a reminder to yourself. Tissue is multiple cells of the same type working together to do some kind of job. Okay, so a tissue is multiple cells of the same type working together to do some kind of a job. As this slide points out, even though we didn't go over it, we have only four main types of tissue in our bodies. If you're curious, you can ask me about them. I'll fill you in on some little itty bitty details. I'm sure, especially looking at a couple of these, like everyone probably has a feel for what muscles are doing. We all, I think, have a general sense about what nervous tissue does. It sends signals all over the body, helps us figure out what's going on, etc. What we're seeing here is just a reminder about definitions we've gotten in the past. Going back to that figure where we would have scaled it up from cell to tissue to organ up to organ system. To jump from the level of tissue to the level of organ, really what you're doing is, by definition, you're combining multiple types of tissue. So if you picked one organ to think about, like maybe the stomach, you're gonna find every single kind of tissue within that one organ, right? Definition is you have to have at least two. In reality, you almost always, I don't know of any exceptions, have every one of those kinds of tissues within one organ. Our stomach has a whole bunch of muscular layers that help us squish around the food and digest it. There are nerve endings all over the place to help us keep track of how the process is going. Those different layers are connected with connective tissue to kind of bind it together. And epithelial tissue is lining the whole inside surface of your stomach. So even just looking at one organ, you gotta combine like all of those tissue types to make one functional organ. As I've said many times before, every organ has specific jobs it's doing, specific, process that it's contributing to. But no one organ can do everything by itself. This is why they work together in groups, okay, called organ systems. Organ systems represent multiple organs kind of coordinating to accomplish some major job in the body, some major life process that needs to occur. They're gonna essentially tag team it. Like, I'll do this part of it, pass the baton to you, you finish this job. That's one way you can think about it. What the book gives us in this chapter, chapter 31, is kind of a brief overview of some of the major organ systems we have in our bodies, shown on the right, and what big life process they are a part of on the left. Now, for me, I'm not gonna read this whole thing to you and talk in depth about it, but I feel like this would be a super easy, I could make a matching section out of this, in a piece of cake for the test. What I want to do is point out two things. Number one, in just a second, I'm going to point out some of the processes that relate to chapters we've already done or will be doing. Number two, if this is the kind of thing that you're interested in, like how different tissues work, what different organ systems do, take anatomy and physiology if you haven't yet. That is the main focus of that class. In this class, it's not the main focus, which is why we're not going to go into every single organ system and what it does. I picked one for us to do in chapter 34. The one that we're going to look at in more detail is our digestive system. And if you look at where this is listed, you probably should have figured out that's what our digestive system is helping us with, right? We eat food and we poop out whatever we can't use. Everyone knows that, right? It's a body process we do every day. So you can find familiar things like that if you look up here. The other one I want to point out, respiratory system, okay? Why does that relate to something we've talked about? Because we've done cellular respiration. You know why the gases we're exchanging are being exchanged. We get oxygen from the air, deliver it to all of our cells to use as the final electron acceptor, and then we carbon dioxide exhale to get rid of a waste product as a result of breaking down the sugars that we took in through our food, right? How do we get the sugars from our digestive system delivered to the cells all over our body? That's what our bloodstream is doing. Our blood is really being used to transport important materials like glucose, oxygen, carbon dioxide, whatever, to all the places in our body that we either need to get it from or need to get it to. That's what this transport system is all about. And again, you can find some other stuff up here that you're familiar with, or just from like living your life to make the connection about what your different systems are doing. Those are always the couple that I point out. The digestive system, because we're going to do the chapter on it in a minute. The respiratory system, because it relates to us thinking about cellular respiration and making a connection to how do we get the reactants into our body, into our cells. And the cardiovascular system, because blood is used to transport all those materials. We are going to look at components of almost all of those systems as well in our rats when we do that dissection starting on Monday. So I think this is a good sort of precursor to lead us into the lab, which is really the only reason I'm going to show you this figure, which I always ask at least one test question about on exam 3, always, okay? Why I like to show this is specifically to get us ready for the rat dissection, because some of these terms are used all over the place in that lab book, to help us realize like what part of the rat we should be looking at. These terms on the side that describe different body cavities, these are really used to help us figure out where we should be looking in the body or where our organs are located. The main two body cavities are dorsal and ventral. Dorsal, think like a dorsal fin, so that helps me remember dorsal is on your back. Ventral, which is where most of our organs are, that's the cavity on the front side of our body, like in front of your spine. The dorsal cavity really just contains two body cavities, the cranial cavity, which is where your brain is, and the vertebral cavity, which is where your spinal cord is. Again, those are the only two things that are on the back of your body in the dorsal cavity. On the front side, in the ventral cavity, there's more going on here. Because it's a bigger area, we divide it into 3 different zones. The thoracic cavity is your chest cavity, right? So what's really there, the big stuff? Your heart and lungs. So if you ever watch, like medical shows on TV or whatever, they'll call someone a cardiothoracic surgeon. Cardio for heart, thoracic for chest cavity. Abdominal cavity is major digestive organs. And I'm going to point out right here, see this separation between those two cavities, the thoracic and the abdominal? There's this line right here. That's a muscle called the diaphragm. It's a thin layer of muscle that is just below your lungs and just above your stomach and your liver. And we're going to find that in the rat to help us distinguish where those two cavities are. The last cavity within the ventral cavity, so we're still on the front side, is the pelvic cavity, which is where reproductive organs are found. And again, the main reason I wanted to point these out to you is, number one, guaranteed there's going to be a test question about it on exam 3, at least one. And number 2, especially these terms, dorsal and ventral, are all over the place in the rat lab. There are a lot of figures in that book that we're going to have to reference while we do our dissection, and almost all of them are labeled dorsal or ventral. It's basically telling you what side of your rat you should be looking at, so I'll make sure I review those terms with you guys in lab next week. The last topic in this chapter, which I think maybe we'll take our break first, and then we'll tackle the big topic we're looking at, is homeostasis, which you heard a little bit about in chapter 1. So we're going to take our break, like a 9-minute break. It's 10:51, so we'll be back at 11. We'll finish off chapter 31 pretty quickly, and then get right into chapter 34 on the digestive system. All right. Hey, how's it going?
Good. We're going on vacation on April 3rd, and we have another test that day.
Okay.
So we're just wondering what our options are
Um,
for taking it.
Yeah, so when are you going to be back?
Well, we're leaving on the 2nd, which is the
Okay.
day before the test.
Okay. I'm just going to get a calendar.
So
So,
you have a
yeah, like everyone's
you have a
allowed.
Tuesday, Thursday class.
I do.
We were wondering if we could maybe
Take it
early
with
that
them.
day.
So you don't have to try to study on vacation. That would be good.
Or yeah.
I'm trying to.
Will they take it
cause-
on?
They might take it on Thursday, so you might be gone already. Yeah. And what day are you going to get back?
We're getting back.
Monday, the next Monday.
Oh, the next Monday? Oh, you're just going for the weekend, basically. Well,
Or it's
long
basically
long
Tuesday or
weekend.
Sunday.
Yeah. So everyone's allowed one makeup. So technically, I usually tell people like going on vacation, but whatever, right? Whatever. So I can take your exams to the Learning Center
I
for
was
you.
wondering that.
Yeah, and you can take them if you want to take them early, so you don't have to try to study while you're gone. I'm
Yeah,
fine with
I would
that.
love that.
I'm fine with that. I can take yours over there like the day before.
Okay, I was gonna say, we could leave it, we could come here in the morning on Tuesday and then.
Yeah, I can make sure I get it there Monday evening, and then I'll
Okay.
have it for you Tuesday. Or if you
Or could we
are
do it
stressed
Monday
about
after
it,
class?
yeah, you could. Yeah,
No,
that'd
I
probably be
understand.
fine. But then, like, we're gonna learn new material on
Yeah,
that
I
day,
know,
so
it's
then you
gonna be
wouldn't have
hard.
time. Yeah, I'd also be fine with it if you want to take it when you get back. So I don't know
personally
what
wanna take it before, but-
beforehand. Yeah, and we can figure that out. Like,
Closer.
well, yeah, next week, a couple weeks beforehand, we can talk about it. Whatever.
Okay.
Yeah. So that's cool. That's
Okay,
fine.
cool.
We'll work it out.
Do we have a lab always a pre-quiz?
I think we do.
It will be on Monday if we have one.
Okay.
You mean that?
We'll be here that Monday.
Yeah,
and
so
we leave
you
on
should,
Tuesday.
and I would have to look at the schedule to see if
So I
there's a
think I
lab
have the
review
thing.
quiz.
Yeah, I think we might have. a
I
quiz
have,
that day.
okay, well, we'll have to figure that out too then. Yeah.
Yeah, because it would be April 3rd.
Is there one?
Yeah, I think so.
Oh, if it's just a pre-quiz, I mean, you can't make those up anyways,
Yeah,
and they're
no.
just 3 points, so I wouldn't worry too much about it.
Okay, thank you.
Okay,
Thanks.
yep, all right. Okay, guys, we're going to get back at it. First things first, any lab questions before we get back into lecture? Okay, I'm going to try to, mental reminder to myself, stop about 5 minutes earlier than I normally would because I want to scurry over to lab, double check the setup. I left very detailed instructions about what I wanted, but I want to double check everything, check the focus on the scopes, all of that, before I open the doors and let you guys in. So try to make sure I stop at about 1:45 instead of 10:02. What we've got left to tackle here at the very end of chapter 31 is homeostasis. And why I said just before break that this should sound hopefully at least a little bit familiar is homeostasis was on that list of requirements for life that we heard about in chapter 1, meaning all organisms on the planet have to be able to do this somehow. So reminder, homeostasis is what I always talk about as sort of maintaining the sweet spot. for any given condition you can think of, and probably some you've never thought about, we essentially have to be in a certain range in order to be alive. The sweet spot, right? If you get too far outside that range on either side, you could die. And so to me, always it makes a lot of sense when I think about it in that way. If it truly is a matter of life and death, which it is, we don't want to leave it to chance and just like hope for the best. So it makes sense to me in that regard that we would have dedicated systems in our body that are really just there to make sure we're constantly checking in on where we're at and making any adjustments that might be necessary to make sure we don't get too far outside that range. At the end of the day, this is really what all of our organ systems are involved with. Every single one of them is involved in some aspect of helping us maintain homeostasis. Couple examples. Respiratory system. We already talked about that a couple minutes ago, right? We breathe in oxygen so that we'll have a final electron acceptor. We breathe out CO2 to get rid of a waste product, right? Being able to have oxygen levels at a certain amount in our body is absolutely required to keep us alive, as you very well know, right? If we don't have oxygen, if we have no final electron acceptor, our cells will switch in a last ditch effort to fermentation, but that won't help us survive. So after a couple of minutes, we'd be done, right? Another one I like to talk about a little bit is blood sugar levels. I always talk about this. I probably did it in chapter 1 too, because most of us probably know someone who's diabetic. So I always say, if you're not diabetic or you don't live with someone who's diabetic, you've probably not thought twice about your blood sugar levels. You probably never have thought about it, because why would you, right? If this whole process is working the way it's supposed to, you would never even think about it. You'd never notice it because everything's running great. But usually when there's some kind of glitch in the system, that's when you notice it. Our blood sugar levels are one of those conditions that we have to be within a certain range. Essentially, if you get too low, you can go into a coma and die. If your blood sugar gets crazy high, way too high, especially consistently way, way, way too high, you can essentially go into a coma and die that way too. So we have dedicated systems to help us make sure we're in the sweet spot, right? The liver and the pancreas are part of this equation to how we regulate our blood sugar levels. And we're not going to go into details about it. I just always try to mention that as an example, because there are so many people who know someone or might be actually themselves diabetic. How our body responds to a lot of this is through the use of hormones. We haven't really talked much about hormones in here. If you're not familiar, hormones are essentially like little chemical messengers. They're made in one place in your body, and they enter the bloodstream and circulate everywhere to quickly deliver a message about what to do or how to respond to a given situation. Ultimately, of course, just like everything else in our body, this comes down to the nervous system. Can we pick up on what is going on and make a decision about what the appropriate response would be? So clearly, homeostasis is really important for us to remain alive. And what we're going to add in this chapter, all of that, we kind of mentioned in chapter 1 in the overview, right, in the list of requirements for life. The new stuff we're going to add in here is how do we do this? How do we make sure that we're remaining in the range we need to be in? How is homeostasis regulated or controlled? Not just the fact that it exists, but how do we keep tabs on what's going on? There are 2 main types of regulation for homeostasis. Negative feedback regulation, which is by far the most common, and there's also at least a couple examples of positive feedback regulation. Here's kind of the difference between them in a first crack. With negative feedback regulation, you're going to be making a change in the opposite direction. Like if you get too cold, your body tries to warm you back up. Or if you get too hot, your body tries to cool you back down. You're trying to counteract, however you're out of balance. With positive feedback regulation, it's change more and more and more in the same direction. There really is only one good example that I've ever heard about. So that's the one that is in our book. It's the one that I will go over with you guys. Like I said, most homeostasis regulation, almost all of it, is negative feedback. Whatever type of regulation it is, there are 2 main components involved in helping us figure out what we should be doing here. First main component is called the sensor, which is doing exactly what it sounds like. This is the part of our body that's picking up on current conditions. Like what's happening right now, where are we at right now, and that information is then sent to the control center. The control center is like the decision-making part. It's going to compare our current conditions to where we should be and make sure they're in agreement. Are we where we should be? Great. If we're not where we should be, we're going to make a decision about how to make a change to get us back to where we should be. How do we know where we should be? Something called the set point. The set point is our goal. That's your range, that's your sweet spot. That's the right place that our body should be for whatever condition we're looking at. So what we're going to do is look at an example of how negative feedback regulation works, see how a sensor picks up on where we're at, see how the control center makes an adjustment when necessary. And before we get to a biology example, we're going to do a non-biology example, which I think is perfect to get us used to this idea. And the example is one that I'm sure everyone is familiar with, at least in some regard. We're going to think about how the furnace works at your house. Okay, so what happens? Well, on your thermostat, you set whatever you want the temperature to be, right? So in this example, we have put our thermostat at 68 degrees, okay? Up top there as well, 68 degrees set point. So you have manually put whatever you want the temperature in your house to be. And the thermostat and the furnace are then working together to make sure that temperature is maintained. I'm going to come down here first since it's winter. I think this is maybe easier to relate to. Sensor in your house is going to pick up what the temperature in your house actually is. If it is 66 degrees in there right now, that information from the sensor is sent to the control center, okay? The control center is basically asking, is where we're at right now equal to the set point? Are we at 68 degrees? No, we're not there. We're too cold right now. All right, if we're too cold, how are we supposed to get back up to 68? We gotta make some more heat here. So that would trigger your furnace to turn on and start putting out more heat until you warm back up to 68. Meanwhile, the sensor is picking up on current conditions always, right? So if it's at 67, 67 is still not 68, the furnace would stay on. Once it gets to 68, now where we're supposed to be, the furnace would turn off, okay? Until if it's super cold out, the temperature drifts back down, the furnace comes back on. Up top here, we're seeing a change in the other direction. If it were like 100 degrees outside, your house is gonna get hotter than 68, I'm guessing, right? I wish they had written this example a little bit differently. I think it could have been a little bit better. All they're showing us is that if it's too hot, your furnace does not come on, like, okay, obviously. I wish what they had done is put like an air conditioning thing here. Like, okay, if it's too hot, then the air would come on until it gets back down to 68, and then you would be fine. Again, the main idea here is it's negative feedback because you're initiating a change purposefully in the opposite direction. If the house is too cold, the furnace comes on to heat it up, or if the house gets too hot, the air would kick in to turn it down. Any questions on how a thermostat works? It's just so weird. Okay. Good news is if you get that, you already know how this works in your body, because body temperature regulation occurs in pretty much exactly the same way. We don't have a little thermostat and a little furnace. Both the sensor and the control center are in a part of our brain called the hypothalamus. And what we're going to do is basically look at the same figure again on the next slide, but now with the biological components thrown in there instead of like the furnace and the thermostat. I think it is important to right away recognize for body temperature, what is our set point? Yeah, 98.6, yeah, that's I think average for everyone. Yours might be a little higher or a little lower. We maybe all have our own personal set point, but average 98.6 Fahrenheit or 37 degrees Celsius. So that's what our hypothalamus has sort of been set to. That's our internal check. And then we're constantly just picking up, where are we at? Are we at 98.6? Are we too hot? Are we too cold? How do we fix this? Remember why this is so important. If you get too cold, you can freeze to death. If you get too hot, you can also die. This is a matter of life and death, so we want to make sure we have it handled. And this is what the hypothalamus helps us do. So I'm going to go through this the same way I did on the last slide. I'm going to start down here with we're getting too cold. Okay? So we know we're supposed to be at 98.6. Our sensor in the hypothalamus constantly monitoring our temperature. So let's say on a frigid day like today, you've been outside for an hour with no coat on. So you're probably a little bit chilly, right? If your brain picks up on the fact that your body temperature is getting a little bit too chilly, there should be a response to try to get you to warm back up. What would that appropriate response look like? I want to explain a little bit about what's written here. Okay, the first thing they have written is blood vessels constrict, and in the background what you're seeing is a picture of skin. How are you supposed to know that's skin? I'm telling you it's skin, that's how you would know. Otherwise, an earlier part of chapter 31 that we totally skipped was never in the notes, looks at skin as an example of an organ, so they pulled that from the early part of the chapter. It's a picture of skin. So specifically, we're thinking that the blood vessels in the skin, which is of course covering the surface of your body, those blood vessels constrict or get smaller. Why might that be a useful response? Well, the surface area of your body, your skin, is clearly where we're interacting with the environment. And if your body is getting cold, you're likely in a cold environment. So if you can kind of shunt the blood flow away from the surface, it gives the heat less of a chance to escape. This is sometimes why people's fingers turn white or kind of bluish if they're cold. Or also think about, where do people get frostbite first? If they're gonna get frostbite. Fingers, toes, tips of your nose, ears. Those are all places, this is gonna sound weird to say, but that are disposable in terms of your survival. If you had frostbite and you had to lose a couple fingers or a toe, could you still survive? Yeah, you'd have to make some adjustments obviously, but you'd survive. Versus if in the core of your body, your vital organs got too cold and shut down, could you survive without them? No. So this idea of blood vessels near the surface getting smaller, what we're really doing is we're conserving whatever warmth we have to protect what's most important, our vital organs. We route blood flow away from the surface and keep it in the interior where it matters. Now the other thing that's written here is strange to me. Sweat glands are inactive. Yeah, okay, when you're freezing, you're not sweating. We got it. That's not helpful to me. What I wish they had written there instead was shivering. Wish they had written shivering, right? When you're really cold and you start shivering, that muscle movement, the friction associated with it actually helps generate body heat, and that can help warm you back up as well. So again, the main idea here is we got too cold and our body is initiating a response that should help us warm back up to get back up to 98.6. Up above, we're again seeing the opposite. What happens when we get overheated? What happens when our body temperature is too high? Well, again, the hypothalamus picks up on that and sends some messages to the rest of our body about what we should be doing. Blood vessels, again, near the surface, dilate or get bigger. In this case, we're sending more blood to the surface so that more heat can escape in that way. This is why sometimes if you're overheated, you get flushed or people get really rosy cheeks. That's because there's more blood flow at the surface so that some of that heat can escape. Here, sweat glands are active because sweating helps you cool off. Right? So again, the main idea with negative feedback regulation is we're initiating a change in the opposite direction. If you get too cold, your body tries to warm you back up. If your body gets too hot, you try to cool back down to bring you back to whatever the set point should be. Any questions on how negative feedback regulation works? Okay, one quick example of positive feedback regulation. Again, this occurs when there's change more and more and more in the same direction. This is much harder to find a good example of, though only one I've really ever seen mentioned is the one our book gives us, which is the dilation of the cervix during childbirth. If you're not that familiar with the cervix, it's usually pretty little, but in order for the baby to pass through and be born, it has to get big enough to allow the baby to pass through, right? You always see it on TV, you gotta get to 10 centimeters. Go ahead, Patrick.
About the cold thing.
Yeah.
There's a survival fatigue that everyone has, that when you get extreme cold,
Yeah.
to the point of almost dying.
Mm-hmm.
There's this moment when your body just overheats to the point that almost you just take everything off because you're so hot. It's like a last.
Yeah. Well, I think as you start getting really cold, kind of delirium and confusion sets in.
It's like a desperate, it's like your body's way of desperately trying to get warm again.
Yeah. That's interesting because that's the opposite reaction that you should hope for, right? Yeah.
I learned that while watching Mount Everest.
Oh, that's cool. That's really interesting. So I would think if you're with someone in the cold and they start doing that, you should definitely be concerned then. Yeah.
It was a survivor's.
Like a recount?
He said he wrote everything because all of a sudden it just felt like this
Super hot.
this insane wave of just heat.
That's strange. That's
Body just became one all of a sudden.
that's really interesting.
Explain it.
Yeah, I certainly can't explain it. Yeah, that's really cool. I know, I like those kinds of stories where it's like, I don't know, not near-death experience. Well, in that case, maybe it was, but I think it's really interesting to see what our bodies can do when they're pushed to the extreme sometimes. It's maybe not what you would expect. All right, so back to what happens during childbirth. The cervix, which is usually small, has to get bigger and bigger and bigger until it's big enough for the baby to be born. Now, to be clear, this is a positive feedback regulation within a window, right? Like once the baby's born, it doesn't keep getting bigger and bigger. Then the cervix shrinks back down. So it's only an appropriate response during labor and delivery. How does our body know what to do? Again, it comes back to hormones. The hormone oxytocin stimulates contractions of the uterus, which helps make the cervix get large enough for the baby to pass through. Because, again, the cervix is getting bigger, bigger, bigger. That's why this is a limited example of positive feedback. What I want to point out is that this kind of response, more and more and more in the same direction, would be harmful if it happens in a system that should be controlled by negative feedback. Right? So we were just looking at body temperature, which if you get too warm, you should cool down. If your body cannot cool you down, if instead you're getting hotter and hotter and hotter, that's an inappropriate response. And if your body can't counteract it, that can eventually kill you, as I've said. This is why if you have a super high fever, you do not want to mess with that. Like if you're, I mean, this is ridiculously high. Usually you hear like 105, 106 for an adult. If you're taking Tylenol and Motrin and that's not coming down, you need to go to the ER. Because if you get too hot, you guys know what can happen. We boiled the amylase in lab, right, and then it stopped working. If you get too warm, the proteins and enzymes in your body can start to fall apart, and then you can't do any of the work required to keep you alive. So again, if this kind of response more and more and more in the same direction happens in a system it shouldn't be happening in, then it could be very problematic, of course. Any questions about the little tiny bit of chapter 31 we did, looking at some of the functions of the major organ systems in our body, looking at different body cavities, and then talking about homeostasis? Okay, so call chapter 31 done, and we'll move on to the next, which is chapter 34. Chapter 34 is all about the digestive system. And I want to make a point, that's why I noted it on the slide as well. This is the only part of this chapter I always do. It's not that I've cut anything out because we're short. This one I'm keeping pretty much intact to how I usually teach it. But I never teach the whole entire chapter. What you would see in the book if you look at this chapter, there's some front material where it basically goes over like the different kinds of digestive systems that different organisms have. Almost taking it back to like the idea of taxonomy, what do different phylums and whatever like have. We're gonna just focus on what we have, which in a nice way is almost exactly what the rats have. Our digestive systems are very, very, very similar. There are a couple differences that I'll talk about when we go over it in lab. But other than that, everything we learn here is gonna prime you guys really nicely for looking at those rats next week, okay? So, if I were to ask you, just, you know, without putting this slide up here, if I were to just say like, hey, what does your digestive system do? I'm guessing that we could have written this list together, right? What does our digestive system do? Well, we eat food, we break it up into little pieces that our body can absorb, and whatever we don't absorb to use, we poop out. Like, we all know how the system works. We've lived with it our entire life. What we're gonna do in this chapter is really kind of break this down into detail and talk about what part of this process happens in what organ of the digestive system. We have what's called a complete digestive tract. That's that one thing I pulled from the early part of the chapter that I'm really skipping. All that term means is that we've got two openings, one at the front end, one at the back end. Not all organisms have that. Some only have one opening that's both entry and exit. So I think that's, you know, a good definition to get out of the way. I don't think I've ever asked anyone about that definition, but I somehow still feel the need to include it just so we all know what it means. How we're honestly going to tackle this chapter is to essentially follow the path of food from the moment we take a bite and start chewing it all the way till the end of the system through what are called the major organs of digestion that I have listed on the top of the slide. if something is considered a major organ of digestion, that really just means the food is going to pass through it at some point. What I've got listed at the bottom are some additional organs that play a role in the digestive process, even though the food doesn't ever go through them. That's why these are called accessory organs. They're contributing to the process even though the food doesn't pass through them. And the three big ones that we're going to look at later in the chapter are the liver, the gallbladder, and the pancreas. And we will specify like what is each of those accessories doing that's so important to the digestive process taking place in the major digestive organs. Before we start looking at where everything happens, I want to point out a couple of important details about how digestion happens kind of throughout this system. The first point I want to make is that digestion in our bodies occurs extracellularly. Remember what that means? Extracellular just means it takes place outside of cells. You guys know how materials are brought into the cell, right? That was a big focus of chapter 5. You know it's harder to bring big, bulky molecules in. Our cells have a much easier time taking in a little molecule compared to a much larger one. That's why most of our digestion takes place outside the cell. So before we even bring stuff into the cell, we're going to see a whole lot of chopping things into little pieces. I'm going to reference chapter 3 a lot throughout chapter 34. Remember, chapter 3 is where we learned how to build the big molecules. And we also learned that you could do hydrolysis reactions and break them apart. Our digestive system is essentially doing a whole series of hydrolysis reactions to take the food molecules that we eat and break them down into smaller pieces. Okay? And that mostly happens outside the cell. Once we get the little pieces, that's what our cells in our intestine can more easily absorb. Digestion really takes place in two different stages or phases. Mechanical digestion kind of just means we're physically breaking food into smaller pieces. And honestly, if you're picturing like eating an apple, the moment you take a bite of the apple, it's already smaller than the whole, right? And then when you do, you chew it, you mash it up into much smaller pieces. That's mechanical digestion, physically busting it up into smaller pieces. That process mainly we think about with chewing, but even after you swallow that food and it gets to your stomach, the stomach continues to kind of churn it and smush it around. So mechanical digestion does continue past chewing or beyond chewing. That's just the main way we think about it. You can only take mechanical digestion so far, right? You can go from a big chunk of an apple to a mashed up apple, but you haven't really touched the molecules in the apple yet, which is why we come in with this second branch of digestion called chemical digestion. This is going to be more in line with what I mentioned a minute ago, tying back from chapter 3. Chemical digestion really requires the use of specific enzymes to perform those hydrolysis reactions that I mentioned. This is, if I had to guess, this is what some of the new material in the chapter is going to be for many of you, because we're going to go over the enzymes that we use to break down like big carbohydrates, big lipids, big proteins in the foods that we eat. We'll learn what these enzymes are called, what reactions they're helping us do, where they're made in the digestive system. That's how we're really going to track all of the digestion that's happening so that our cells are handed the littlest molecules that are easiest to bring in. So that's what we'll be tracking throughout chapter 34. True to form, we've got a big intro figure right up front in the chapter. I like this figure. I think it's a good one. I'm not going to walk you through it. I feel like that would just be super boring because you can all read it on your own. But I wanted to point out that it's here, and I wanted to make sure to put it in the lecture notes so that even if you don't have a book, you still have access to this figure. I like it because it shows us a big overview, like what do the organs look like and where are they in relationship to each other. But it's also nice because next to each organ, they've listed a little recap of what are the things going on in that organ. So it's a nice figure to come back to, maybe more so as a review, to put it all back together again at the end once we've been over it. What I'd rather do is just start getting right into it, okay? Remember, we're going to start from the moment you take a bite of food. So the first organ we're looking at is your mouth, where both types of digestion begin. I think that's the big point I want to make about the mouth. As soon as you take a bite of food, we immediately start both prongs or aspects of digestion. Mechanical for sure, because you're chewing the food up, you're mashing it into smaller pieces. But chemical digestion also begins here, because in our saliva, we make an enzyme that you should be familiar with, right? What does amylase do? Breaks down starch, starch hydrolysis. Why do I expect that you should know that? We did a whole lab on it. It's on the practical today. Remember, amylase takes a huge starch, which is a big sugar, and chops it up into smaller sugars. And we make that enzyme in our saliva. So as you're chewing your food, if you eat anything that has starch in it, remember, starch is made by plants. You eat any kind of plant material like that apple, or like a cracker, or a piece of bread, the amylase in your saliva is immediately getting started on chopping it into smaller sugars, the sugar maltose, which we talked about in lab. Kind of a, I don't know, weird or cool way you can think about this. If you have like a saltine, if you have saltine crackers at home, eat one later and chew it for way longer than you would normally chew it. Like, don't just chew it and then swallow it. Keep chewing it, keep chewing it, keep chewing it. If you chew it for long enough and you give this amylase long enough to work, it'll start to taste sweet to you. Because if enough starch is broken down, it'll convert to maltose, which is a simple sugar that tastes sweet to us. So that's kind of a weird, a weird thing you can try later if you want to make that connection. In this figure, there's a lot labeled over here. I really just want to point out kind of 2 or 3 things, honestly. One is this big brown blob right here, bolus. Bolus just means chewed food, right? That's what I have defined over here. Bolus is the food that you have chewed up that has mixed with saliva. I don't know why they drew it brown over there because it looks like poop, but it's chewed food that you're swallowing. We are going to follow that food. We're going to follow it because it's got to go somewhere else. It's going to go into the next organ we're going to look at, which is the esophagus. The esophagus is a little tube in your throat that connects your mouth to your stomach. So to get the food to the stomach, the bolus has to pass through the esophagus. And I want to point out there's another tube in your throat, in your neck. There's a trachea there as well. Trachea is the tube that carries air down to your lungs. So you have 2 tubes in your throat, one that should take food to the stomach and one that should take air to the lungs. And we want to make sure we're routing things properly. That's what this little flap right here is all about. This flap called the epiglottis. It's like a little flap of tissue that when you swallow food, it's forced down and it covers the opening to the trachea. This is how we're making sure that our food or water doesn't sneak down into our lungs, which would cause problems, right? If you've ever heard the expression like it went down the wrong pipe, like if you're eating or drinking and you laugh or cough or whatever, like it can make you choke a little bit, that kind of really means it went down the wrong pipe. If you're laughing or exhaling at the same time you're trying to swallow, this flap can like stutter out of position, and this bolus can start to kind of trickle down into the trachea, which usually results in violent coughing when you start hacking up a lung. Why? That's your body trying to clear that debris out of your airway so it doesn't suffocate you, essentially, right? So you cough it up and you hope then it can go down the correct pipe. Any questions on bolus, epiglottis, esophagus, which we're going to next, or the trachea, which goes to the lungs? Okay, we're going to look for both of those in our rat, and I'll say this probably in lab next week too, but those 2 tubes in your throat, they are right on top of each other. The trachea is on top and the esophagus is underneath. So when you're looking for the esophagus, you'll kind of have to push the trachea out of the way a bit, and I'll of course show you guys how to do that. Again, what we're going to do is follow that bolus down into the esophagus. Once again, the esophagus is just a tube that connects the mouth to the stomach, okay? What I want to do is talk a little bit about this structure that you're seeing in the picture, because it's not only found in the esophagus, essentially some version of it is found throughout the rest of the digestive system. So I think it warrants taking a quick peek at it. I'm not going to spend as long here as I normally would because we skipped the part of chapter 31 where we would have talked about what these different tissues are, but I at least want to point out there are different types of tissue here. We've got muscle tissue here, we've got a lining that's covering the whole inside surface. There is a lot going on even just with this one organ. Remember, to make an organ, by definition, you have to combine multiple tissue types, and I think this view is a very nice way of recognizing that. So once we swallow and the bolus, pushes the epiglottis to cover the trachea and we route the food into the esophagus, there's a very particular way that it's going to move through the esophagus, and that's what I have written on the next slide. But before I look at that with you guys, I want to make the point that at the bottom of the esophagus, there is what's called the lower gastroesophageal sphincter. Sphincter just means it's a muscle wrapped around a tube. The tube is the esophagus. There's a muscle around it, right? So when that muscle contracts, it closes the tube. When the muscle is relaxed, the tube is open and food can flow through it. Now this is like a big scary looking name, lower gastroesophageal sphincter. It's just describing to you where this particular sphincter is. It's at the bottom, lower, end of the esophagus as it dumps into the stomach. It's telling you where it is. It's like a little doorway at the very bottom of the esophagus. A normal position for the door is to be closed. So that muscle is usually tightened to close the tube. When the food gets there, it's like knocking on the door and then it's let into the stomach. Okay? It's closed, the food comes, it opens to let the food in, and then it closes again. If that little doorway is open when it shouldn't be, it can cause some issues, which I'll come back and talk about a little bit later when we're talking about the stomach. It'll make more sense then. So what we're going to see with the next slide is the type of movement that's used to carry the food from the mouth through the esophagus down to the stomach. This type of movement is called peristalsis. This really just means there's kind of like a rhythmical, wave-like contraction of the muscle that's surrounding your esophagus. Your entire esophagus is wrapped in layers of muscle. When you swallow your food, are you thinking to yourself, let me squeeze that food down to my stomach? No, this is muscle that kind of works without you even realizing it's happening. It's a particular kind of muscle called smooth muscle. And this figure to me is a little bit strange. I wish they had tilted it so it will be going from the top, like where the mouth was, actually up top, and then showing you down below where the stomach would be. All this is trying to get at is how peristalsis works. So again, this whole thing is wrapped with layers of muscle. Right behind where the bolus is, we squeeze the muscle and it pushes it forward just a bit. We then relax that muscle and squeeze just behind where it is to push it forward a bit more. That's why it's like rhythmical. You're squeezing just behind the bolus all the way down. That's what peristalsis looks like. That type of movement is used to move the bolus through the esophagus. That very same idea of movement is used to move the digested food later through the intestine. It's a very similar kind of movement. Any questions on what peristalsis is before we hit the stomach? All right, stomach. It always seems weird to call it like a J-shaped organ, but it kind of is. You know, if you squint a little bit, you can see it looks like the letter J. It's often tilted a bit more in our bodies though, so it's a little bit sideways. First thing I want to point out here, because the book for some reason did not label it, so I added a label for you. Right up top where that arrow, that blue arrow ends, that's the location of that lower gastroesophageal sphincter. Right? So you chew your food, you swallow the bolus, it gets pushed down through peristalsis through the esophagus, then it hits that lower gastroesophageal sphincter. Remember, picture like knocking on the door and being let into the stomach. The stomach is also wrapped in layers of muscle, and the inside lining, see all these little bumps that are in here? Those are structures called rugae. I'm only mentioning that because it's going to be in the summary video I'm going to show you at the end of chapter 34, and I didn't want that to be the only time you heard it. Not a critical term, it just means the bumps on the inside of the stomach. Why I'm mentioning them is because remember I said earlier, mechanical digestion continues in the stomach. So once the bolus is in there, it's going to continue to be mashed as the stomach muscles are kind of squeezing it around, and it's going to rub up against those bumps and be mashed further apart. So that's, continued mechanical digestion. Big things that you will find in the stomach, important properties of the stomach, like what's being made there to help us digest our food. Two big things: gastric acid, which if you've taken chemistry before, you might recognize hydrochloric acid, a strong acid, and we're making pepsin, which is an enzyme. I'm going to say more about that enzyme in just a second. Okay? If there's acid being made here, you should not be surprised to see a low pH. average pH of the stomach, due to all that acid, is around 2, which of course is quite acidic, right? Neutral is 7, and every number you decrease from that is a factor of 10 that you're changing it by. So the stomach is quite acidic. Let's talk about amylase for a second, okay? Amylase, that you know chops starch into little pieces. It was in saliva, so as soon as we took a bite of food, we started chopping the starch apart. Do you guys remember testing amylase at different pH values? Did it work at 4? No. It worked best at 7. Good. It worked at neutral. And you tried it at 4, and it didn't really work. Do you think it's going to work at pH 2? No. Why am I making a big deal out of this? Essentially, amylase is working while you're chewing. We don't chew for all that long, right? You swallow it, it gets to your stomach, that amylase is done. It cannot work under these acidic conditions. And I'm saying that specifically to prime us for something we're going to see in a little bit, maybe on Monday actually. We make another version of amylase later to finish the job started by the one in our saliva. Because once it gets to the stomach, it can't function anymore. What does work very well at pH 2 is the protein, the enzyme called pepsin that we make in the stomach. This is an enzyme, so we're doing some more chemical digestion now. What does pepsin do? It kind of does the equivalent of what amylase does to starch, chop, 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 but pepsin does it to proteins. So when you eat a food with protein in it, and that protein gets down to your stomach, pepsin starts chopping it up. Go ahead, Patrick.
I've always wondered about this in the stomach.
Mm-hmm.
The digested food.
Yeah.
Is that like a pool, or is it like the whole stomach?
I think, yeah, technically speaking, I should probably say I'm not sure. The way I picture it is I do not picture our entire stomach filled with acid. I picture it like there's always a little bit in there, and then additional can be secreted if there's a bunch of food in there. So my guess, I'm not sure.
I've seen thousands of pictures of the stomach, but I
Yeah.
don't know how much fluid is.
Yeah, and my guess is it would be hard to tell unless you're seeing an image from like a scope done on someone, because if you're seeing just a picture of a stomach cut open, all the juice would have leaked out, right? So my guess is that there's always a little bit sitting in there to be ready in case we eat something, and then as you're eating something, it would trigger more to be secreted.
Right.
That's my guess. Yeah. I want to talk about this acid too. Like, what does it help us with? How is it related to pepsin? This, I want to talk about proteins and remind you what you know about how proteins are built. Remember, proteins have to fold into that correct 3-dimensional shape in order to be functional. And that shape, because it's held together by hydrogen bonds, which are the weakest kind of bond we learned about, they are sensitive to things like changes in temperature or changes in pH. So again, thinking about protein that might be in the food that you ate, you're chewing the food, the bolus gets to your stomach, and all that acid hits those proteins, and most of them probably unfold, right? Take your 3D pipe cleaner sculpture, and I'm just going to pull the end straight and destroy it. So the proteins are just laid out, and then pepsin says, chop, 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 right? We're now starting chemical digestion of proteins. We started digesting carbohydrates, sugars, in the mouth. We're done with that for the moment because amylase can't work in the stomach, but now we're starting to work on the proteins that are in the food that we ate. All that acid, by the way, that helps us digest our food and protect us from like foodborne pathogens, it presents additional problems. As much as it's helping us, we also have to protect ourselves from it. pH 2 is quite low. It's like corrosive, like acid would be. It is acid. So we have to make sure we're protecting our cells in our stomach from being damaged by that acid. And that's why I've noted here that there's a layer of mucus that's kind of coating the whole inside of your stomach. That sounds pretty gross, and I think, to think about our stomach being coated by mucus sounds disgusting, but that mucus is playing a really important role. It's secreted by these cells lining the stomach, and it just makes like a little barrier layer here. So the acid can be secreted past it, and then it can't like bounce back and burn our own cells. If that mucus layer is eroded for some reason, then you can run into problems because the acid can start to damage your own tissues. Speaking of mucus and acid, did I mention anything about mucus in the esophagus? I didn't, because there is none. Remember I made the point that that little doorway, the lower gastroesophageal sphincter, should be closed. If it's open when it shouldn't be, stomach acid can splash up and burn the bottom of your esophagus. In a limited basis, that would cause heartburn, a burning sensation after you eat certain foods maybe. If that sphincter is open longer term, if it's more of a serious issue, it can lead to severe acid reflux or GERD, and that's a more serious issue. And again, that comes from the lower gastroesophageal sphincter not shutting all the way when there's no food passing through, because the esophagus doesn't have that mucus. So the burning sensation is really the tissue in your esophagus being burned by stomach acid. Now that we've mixed our bolus together with more liquid from the stomach, right? We had the gastric secretions, we've got the acid, we've got the pepsin, it's more liquidy now. So we flag it with a different term, described as chyme. So bolus entered the stomach, it mixed with all the stomach juices, and now it's gonna leave the stomach, being called chyme. There is, just like there was at the top, there's a little doorway at the bottom of the stomach as well, another sphincter, a muscle wrapped around a tube. This one has a different name because it's in a different spot. This one is called the pyloric sphincter. Okay. So let's just do a vocab check, make sure you can follow what I'm saying. The bolus enters the stomach through the lower gastroesophageal sphincter, and then chyme leaves the stomach through the pyloric sphincter. Let me say that one more time to make sure we've got it right. The bolus enters the stomach through the lower gastroesophageal sphincter, and then the chyme leaves the stomach through the pyloric sphincter. Any questions on what's happening in the stomach? Okay, so far we've heard we're starting to break stuff down, but we haven't really absorbed any nutrients yet. That's because we haven't been in the right spot yet. No nutrient absorption really happens until our next organ, the small intestine. First thing I always say about the small intestine is that that name can be slightly misleading. It's not a small organ at all. It's actually quite large. The name comes from the diameter. It's a pretty narrow tube, but it's pretty long. Average length of adult human small intestine, 6 meters long. So, you've seen a meter stick in lab before. Line up 6 of those end to end, and that's about how much small intestine you have inside your body, all coiled up. So that's a lot. And I'm going to talk about why there's a lot and how it helps us to have so much in just a little bit. First, I want to talk about the 2 major things happening in the small intestine, okay? We are going to complete, as in finish, job is done. We're going to complete the act of digestion, which we started in the mouth as soon as we took a bite of food. There was mechanical there from chewing, there was chemical there from amylase. We continued it, both types, in the stomach, where the mashing up of the food was mechanical and the pepsin was chemical. We're gonna finish digesting carbohydrates, which we'd already started, proteins, which we'd already started, and lipids, which we do totally within the small intestine. As we finish breaking things up into these little pieces, this is where we're going to actually absorb those nutrient molecules. All the sugars, all the amino acids, all the little bits we've chopped it into, this is where we actually soak them in and are able to transport them around our bodies to use them. So the small intestine is a really important organ. You cannot absorb the nutrients if you have not completely broken them down. So both aspects here are really crucial to us getting the nutrients we need from our food. That's where I'm going to leave it for today. So on Monday, we'll pick it up by talking more specifically about the small intestine and how we're accomplishing both of those major jobs. Any questions on the end of chapter 9, the little bit of chapter 31 we did, or the beginning of chapter 34? Okay, before you guys pack everything up, or I guess as you guys pack everything up, I have an assignment for you on chapter 9. I just want to give it to you so you can work on it this weekend. As you are looking over chapter 9, remember I said, don't wait till the night before exam 3. You want to be looking at stuff all along. This is something you can look at while you're going over your notes to make sure you're not missing big stuff. Up top here, pictures. The boxes above the pictures, you're going to label with the different steps of the cell cycle. Everyone pick up on that. cell cycle. I did not say mitosis. That means one of the steps. One of the steps is going to be interphase, okay? And the other 4 pictures drop back down to the normal prophase, metaphase, anaphase, telophase. The numbered lines 1 through 10 are different structures you should be labeling, like cell membrane, chromosome, spindle, whatever. And then down at the bottom, there are some questions asking you about different parts of the process or what the outcome might be. So I'm going to treat this like homework. Tackle as much of it as you can this weekend. Do not come back with it blank. We can take a couple minutes and talk about it on Monday, go over it, whatever. Okay? Any questions?
You said one of those is interphase and the rest.
Yep.
are
E is interphase, okay? And then the rest are the steps in mitosis. Good. All right, so I'm going to give you guys all a copy, and then I'm going to scurry over to lab to double check everything. You might get there before me, but I'm going to go in alone first. The doors will still be locked. When I open the door, there will be, I'll make an announcement about like how this is going to go. But essentially, once you're in that classroom, once you're in the lab, your phone cannot be out. It cannot be visible. We have been instructed by the department that if we even see a cell phone, we are to give you a 0. So that actually happened to someone last semester. So don't do that. And then I'll rehash that again and go over directions once we're over there. Did I miss anyone with a copy? Okay, I'm going to scurry over there. Once I've got everything double, triple checked and your Scantrons laid out for you at stations, I'll open the doors and let you guys in. Should be right around 12. All right, I'll see you over there in a couple minutes.
Good luck with today's lesson.